Inside you, right now, flows a piece of genetic code that has puzzled scientists for decades. It's carried by nearly every person on Earth, by every great ape alive today, and by two extinct species of human. Nobody could fully explain why it survived, unbroken, across millions of years of evolution. For years, people online have filled that gap with strange claims about unusual blood types, alien ancestry, hybrid bloodlines out of ancient mythology. Today, we're setting those stories aside. The real story is stranger, older, and much better proven. It doesn't point to outer space, it points to a cave in Siberia, a rabbit experiment from 1940, and a piece of your own DNA that has survived mostly unchanged for millions of years. Here's the number to start with: 85%. That's how much of the human species carries this blood type right now. This comes from researcher Garratti and his team, published in the journal Transfusion in 2004. Turn that percentage into people, and it's enormous. Nearly 6.9 billion human beings out of about 8.1 billion alive today, all carrying the exact same protein on their red blood cells. This one protein decides who can safely get a blood transfusion in an emergency. It also decides whether a pregnancy goes smoothly and who blood banks call a universal donor in a crisis. This is the ancestral baseline of the whole human family. It's the default setting for our entire genus, going back long before modern humans existed. Generation after generation, migration after migration, a rival version of the same gene tried to take its place. It mostly failed. Until now. In 1940, two researchers ran an experiment. They had no idea what it would reveal. Carl Landsteiner and Alexander Wiener were already famous. Landsteiner had discovered the ABO blood types 40 years earlier, the A, B, AB, and O groups you already know. In 1940, working with Wiener, he tried something new. He injected rabbits and guinea pigs with red blood cells taken from a rhesus macaque monkey. They built antibodies to fight off what looked like an invader. Then, Landsteiner and Wiener mixed that rabbit blood serum with human blood samples. Something strange happened. In about 85% of the samples, the blood clumped together instantly. Doctors call this agglutination. In the other 15%, nothing happened. No clumping at all. It was as if the protein simply wasn't there. That gap had a simple explanation. It was the presence or the absence of one single protein built into the outer wall of each red blood cell. ABO blood markers are small sugar tags stuck to the outside of the cell. This protein is different. It's physical scaffolding, built directly into the cell's outer wall. They named the new protein after the monkey that revealed it, the rhesus factor, or RH for short. Positive for the majority, negative for the rest. A year earlier, in 1939, doctors Philip Levine and Rufus Stetson had already traced this exact protein to a mother's dangerous reaction to her husband's blood. For decades, doctors thought this was the same discovery as the monkey experiment. By 1963, researchers proved the two antibodies were actually different. The clinical name RH factor stayed for humans, and the real danger this protein could cause, a mismatch between a mother and her unborn baby, stayed unsolved for almost 30 more years. Then, in 1968, a new injectable treatment finally closed the loop and made that danger preventable. Tucked into the Pyrenees Mountains, on the border of Spain and France, lives a group of people who speak a language unlike any other on Earth, a genuine language isolate. These are the Basques, and genetically, they do something almost nobody else on the planet does. Impossible. Across nearly every population scientists have measured, including sub-Saharan Africa, East Asia, and the Americas, this blood type shows up in 95 to 100% of people. Almost everyone has it. Among the Basques, that number drops all the way down to 65 to 70%. For decades, scientists filed this away as a strange local quirk. New research says otherwise. Researchers Sarno and Giuliani published new research on bioRxiv in 2025. They traced this anomaly back to an ancient group of hunter-gatherers. These people sheltered in the mountains of Spain during the last ice age. They carried an unusually high rate of the genetic deletion responsible for the opposite blood type. Today, that same deletion shows up in 47.2% of the Basque gene pool. The story didn't stop there. A separate study came from researcher Alan Toft and his team in the journal Nature in 2024. They found a second wave layered on top of the first, thousands of years after farmers first arrived from the Near East. A new group called the Yamnaya swept into Western Europe. This happened during the Bronze Age. They came from the steppes near modern-day Russia. Each new wave mixed further with the people already sheltering in that mountain pocket. That mixing concentrated the deletion in exactly this one corner of Europe. The same research links it to other regional traits, like skin color genes. Nowhere else on Earth did this exact chain of events happen the same way. This trait doesn't spread evenly around the world. It clusters unevenly by region, and the pattern breaks down completely in one unexpected place. Sub-Saharan Africa sits at 95 to 99%. This is where the whole system began. East Asia, China, Japan, and Korea climbs even higher to 99.0 to 99.5%. It's so close to universal that the rare opposite type has earned a nickname in Chinese hospitals, panda blood. Doctors treat it like an endangered species. Southeast Asia clears 99% on its own. Indigenous populations across the Americas sit at 99 to 100%. The very first people who crossed into this hemisphere carried almost none of the deletion at all. Oceania and Aboriginal Australia hold above 99% too. They follow the same ancient path that carried modern humans out of Africa 60 to 70,000 years ago. Then the map starts to slide. South Asia, India, and Pakistan drops to a still high 93 to 95%. The Middle East and North Africa dip further, down to somewhere between 88 and 93%. Eastern Europe slips to 84 to 86%. Western and Northern Europe hold around 83 to 85%. Right at the edge of that slow decline, the floor drops out completely. The Basque Country sits at 65 to 70%. That's the lowest rate recorded anywhere on the planet. The synergistic selection model from researchers Sarno and Giuliani points to something else at work: active biological selection favoring survival advantages that had nothing to do with reproduction at all. Here's the rule that should have settled this hundreds of generations ago. In 1942, a geneticist named J.B.S. Haldane worked out the math. When a mismatch between a mother and her child removes the rarer version of a gene, that rarer version should slowly disappear. Every population should end up as one type or the other, no lingering split, no 35% minority holding on for tens of thousands of years in one mountain range. And yet, there it is. The Basque number hasn't drifted towards 0. It hasn't drifted toward 100 either. Something has been actively propping up the losing side of this equation for thousands of years. And for most of the 20th century, nobody could say what that something was. The answer was hiding inside a parasite. Toxoplasma gondii is a tiny parasite. It infects about 1/3 of the entire human population. It forms permanent cysts inside brain tissue. In ancient environments filled with wild cats, its favorite host, getting infected at some point, was almost guaranteed. Researcher Jaroslav Fleger and his team at Charles University spent years studying what this infection does to reaction time and coordination. They found infected people react measurably slower. Their risk of a car accident triples. Their research found something unexpected. This blood type protects against that decline. People who carry it show no real drop in reaction speed, even after infection. People with the opposite blood type declined sharply once infected. Then came a second layer of protection. This one works at the cell level, not the brain. The protein teams up with 3 partner proteins. Together they form a working unit inside the cell wall. This unit acts as a transport channel. It moves ammonia and other waste gases across the cell membrane to help balance your body's chemistry. It also helps move carbon dioxide out of the blood during hard exercise. Researcher Repoche and his team studied this in 2006 in the journal Transfusion Clinique et Biologique. Later work backed it up in the Proceedings of the National Academy of Sciences. Their conclusion: this is a working gas exchange system built right into the cell wall, doing real work every day. Two separate survival advantages stacked on one inherited protein. One protects the brain from a parasite. The other keeps basic cell chemistry running under stress. There's a real cost, too. When a mother without this protein carries a baby who inherited it from the father, her immune system can eventually treat that baby's blood as a threat. That's the exact conflict researchers spent decades trying to understand. Evolution doesn't hold on to something this costly, this risky, and this old without a very good reason. Blood banks use a phrase most people have heard but never think about: universal donor. It sounds like an honor. In real life, it's a supply problem with a human face. The World Health Organization tracks this. So does researcher Garatti in the same study mentioned earlier. Together, they show that 38 to 45% of people carry a specific mix, no A or B markers, plus this exact protein. That mix makes their blood safe to give almost anyone in an emergency. It doesn't matter what type the patient has. Trauma teams depend on this blood to stabilize patients the second they arrive. There's no time to run a full match. This is the blood kept closest to the door, ready to go. And that's the twist hiding under the honor. The same trait that makes this blood useful is the same trait that keeps hospitals needing more of it. Being universal keeps demand high, since supply never really catches up. Trauma teams lean on it hardest for male patients and women past childbearing age during serious blood loss. The whole system depends on regular people showing up to give blood again and again, just to keep up. This story is still written into your body today, in ways you can measure and ways you can't. Start with what you can actually measure. The gene sits on chromosome 1. Scientists label the exact spot 1p34.1 to 1p36. Its frequency around the world lines up almost exactly with the migration map you just heard. High across Africa and Asia, lower across Western Europe. That's the visible, measurable layer. Harder to measure, but just as real, is what's happening underneath the surface. Remember the brain protection from Flieger's research? Steadier reaction times, more stable motor control during a parasite infection. That falls into the invisible category. It's the kind of benefit you'd never notice day to day, unless it suddenly disappeared. In ancient hunting environments filled with predators, that steadiness was the gap between reacting fast enough to survive and not reacting fast enough at all. There's a third layer too, sitting between visible and invisible. It's the everyday reality of being an easy match in almost any medical situation. A routine transfusion, a normal pregnancy. Your body isn't building antibodies against something your own ancestors have carried for millions of years. Millions of years later, natural selection is still running by the same basic rule it always has. It keeps what works. A mystery this old and this widespread was always going to attract wild theories. Some say it must come from outer space, ancient beings mixing with early humans, a marker nobody can trace. Others dress up the same idea in older language. They point to legendary hybrid bloodlines from ancient religious myths. And plenty of online forums push a different belief, that certain bloodlines simply aren't fully human. Seductive stories, but completely unnecessary. Scientists have sequenced whole genomes from thousands of humans, ancient humans, and other primates. That work settles this cleanly. Chimpanzees carry the working version of this gene. So do gorillas, bonobos, orangutans, and rhesus macaques. Neanderthals carried it too. So did Denisovans, a separate ancient human relative we only know about because of DNA. It's the original, complete version of the gene. Our entire branch of humans has carried it for over 2 million years. An even older version of the same gene goes back roughly 40 million years across the whole primate family tree. Everyone alive today inherited it from a shared ancestor deep in that tree. No aliens required. The real answer comes from a 2021 paleogenetic study, and it's better than fiction. In July 2021, researcher Silvana Condemi and her team published a study in the journal PLOS One. Its title, Blood Groups of Neanderthals and Denisova Decrypted. They studied detailed DNA from 3 Neanderthals found in Siberia, Croatia, and the Altai Mountains. They also studied one Denisovan, known as Denisova 3. The Denisova DNA carried a complete, fully working copy of this gene. It's identical to the version still found in sub-Saharan Africa today. That one result tells us something remarkable. The last shared ancestor of Denisovans, Neanderthals, and modern humans already carried this exact blood type. That ancestor lived long before any of these groups split apart. The Neanderthal DNA held something stranger. All 3 Neanderthal females carried a unique partial version of the gene. It was missing a few specific pieces. It sat between the usual positive and negative versions, matching neither. For years, researchers assumed this version had died out along with the Neanderthals. Then, in 2019, geneticists found that exact same version alive today in 2 living people, an indigenous Aboriginal Australian and a person from Papua New Guinea. That match is direct proof of ancient interbreeding. It shows that early modern humans crossed paths with Neanderthals. It likely happened somewhere along the migration route through the Middle East, on the long journey toward Southeast Asia and Australia. A small piece of Neanderthal blood chemistry rode along in the human genome ever since. There's a darker footnote too. Condemi's team links that same partial version to a mother-baby blood conflict, the same kind we've been talking about this whole video. It may have been one of several reasons Neanderthals struggled to survive as their numbers shrank. Modern medicine solved this exact problem in 1968. It may have helped end an entire branch of the human family tens of thousands of years earlier. This is an active, urgent presence in hospitals today. About 85% of the world's population carries it. That's more than 6.8 billion people. Blood banking systems everywhere are built around that fact. The World Health Organization treats it as the backbone of emergency medicine. There's a quiet irony sitting at the center of all of it. The fix for the one real medical risk tied to this protein has existed since 1968. The fix is simple, it's proven, and it still isn't reaching everyone who needs it, everywhere in the world, even now. What you're carrying is a living record. It rode along on every wave of migration that carried modern humans out of Africa and across the whole planet, and it's still doing the same job today. Moving gas across a cell wall, protecting a brain from parasites. Every time a trauma team reaches for the nearest matching bag of blood, that 2-million-year-old inheritance is doing its job again. So the next time someone brings up alien blood or ancient hybrids, you'll know the real answer. Tell us in the comments, did you already know your blood type traces back to Neanderthals and Denisovans? Or is this the first time you're hearing it? Like this video, subscribe, and stay with DNA Detect for more stories hidden inside your own DNA. Your blood type carries a story 2 million years in the making. You're carrying the original.